Hello, my name is Hannah Klatte, and I will be presenting my research in post-catastrophic food resilience. Specifically, we'll be looking at using nutrition from fungal inoculation and enzymatic hydrolysis of willow. So, my aim is to produce a sustainable food source under the limitation of post-catastrophic scenarios, such as a nuclear winter. A model using only 0.3% of the world's nuclear arsenal shows that there would be dramatic changes in environment. So these changes might include a 40% reduction in sunlight, a 10 to 30 degrees Celsius reduction in temperature, and 25 to 50% reduction in precipitation. So these changes in environment can cause a significant stress on agricultural processes and may also threaten food insecurity for many people, or famine as well. So we are interested in creating a creative way of producing human nutrition under these limitations of a post-catastrophic scenario. So what we're looking at is using lignocellulose as a food source. Lignocellulose is commonly available, and it is made 35 to 60% of cell wall polysaccharides, which can be degraded into edible components such as glucose. We're interested in degrading it through enzymatic hydrolysis. Enzymatic hydrolysis are a source of lignocellulosic degrading enzymes that can break down the cellulose into glucose. However, it is also important to pre-treat biomass using various different means, and that will get rid of certain interfering components. So when you treat it with pre-treatments, then you should be able to increase enzymatic hydrolysis. All right, so we are interested specifically in pre-treating our biomass with edible white rot fungi. Some advantages to this is that not only can it degrade some of these interfering components like lignin, but it can also produce its own source of proteinaceous material, which can be very advantageous for our experiment. So first, we inoculated the biomass with either Pleurotus austratus, which is also known as the oyster mushroom, or Lentinula edodes, which is the shiitake mushroom. Then we analyzed the biomass in the beginning of inoculation as well as after fruiting for both of these. And the same biomass substrate was inoculated with the opposite type of species. And we then again looked at the composition of the biomass before and after. In addition, we also took into consideration the fruiting bodies that were produced by these substrate materials as well, because both of these enzymatic hydrolysis and fruiting bodies can produce edible food for human nutrition. So first, let's look at the mushroom yield. The mushroom yield shows that Pleurotus austratus was the only one that actually produced any kinds of mushrooms. Moreover, Pleurotus austratus, when grown on substrate that was only singly inoculated rather than doubly, produced significantly more mushroom biomass. Then let's look at enzymatic hydrolysis. We found that enzymatic hydrolysis was significantly increased in many different treatment groups. For example, all of these treatment groups, besides the two Pleurotus single inoculation, PA early, PA late, those all had significant higher compared to the willow control, which is the light blue. The dark blue is a standard of 100% cellulose that allows us to show how effective the enzymatic hydrolysis can be. Then we considered the nutritional value created by both enzymatic hydrolysis and by the fruiting bodies. So as you can see here, most of our groups besides Pleurotus and early treatment created a significantly higher number of calories per dry substrate. So this shows us that a lot of our treatment groups were very successful in increasing the number of calories that can be obtained per amount of dry substrate. So what would that look like in a post-catastrophic scenario? Well, using this data, about 1.3, which is the most effective treatment group, to 3 kilograms of dry biomass is needed to be able to produce enough food for the daily intake of 2,000 calories per day. So this would mean that you need about 5 to 10 bags of 1 kilogram wet willow substrate to be able to produce the mushrooms and enzymatic hydrolysis of that would be able to give you this nutrition. So these treatment groups using this 1.3 to 3 kilogram dry biomass would be able to provide you with 106% of the daily value of carbohydrate intake 47% of protein intake and 2.6% of lipid intake. So this tells us that this treatment is relatively efficient. However, it is deficient in proteins and lipids. So that is a consideration to take in mind. So that brings us to our final conclusion. The use of a white rot fungi pretreatment system followed by enzymatic hydrolysis is a feasible food resilience strategy in a post-catastrophic situation. Here are my references. So a little bit more about the project. I first joined the Anderson Lab in my sophomore year of college, and I was really interested in the food resilience project because I was interested in nutrition and I wanted to leave some sort of broader impact, and being able to create some sort of response in a catastrophe for food resilience seemed like a really interesting project to me. So that's how I joined it. And I was working on learning enzymatic hydrolysis, things like that, for about a year before Dr. Anderson, who has been my greatest mentor and support in this whole project, and we started to think about using fungi as a source for some of this lignocellulosic degrading components. And we also were interested in what 2 or more fungi would do. So I designed this project independently with his support, and I've been working on it for the last year. And being able to create this project was very interdisciplinary because I had to get some of the willow biomass from Dr. Kamanian, as well as I had to grow the actual fungi in the mushroom research lab with Dr. Pecchia. And then eventually I had to bring it back to our lab to do some of the analysis that you saw in this data. So it was a really big project that I'm really proud of doing, and I'm hoping to continue this research and to eventually have a publication by looking at some of the more time periods in between the whole process. So looking not only at the beginning and end, but I also collected samples all throughout the process. So I want to look at that and hopefully get a publication out of that. So thank you again for listening to this presentation. I hope you learned something new, and I hope you found it interesting. Thank you.